Hello students. Today we'll see about papain extraction. Papain also known as papaya proteinase 1. It is called a cysteine protease enzyme present in the papaya. The botanical name of papaya is Carica papaya and also called mountain papaya. Papain family. Papain belongs to the family of related proteins with a wide variety of activities, including endopeptidases, aminopeptidases, dipeptyl peptidases, and enzymes with both exo and endopeptidase activity. Members of the papain family are widespread, found in baculoviruses, eubacteria, yeast, and practically all protozoas, plants, and mammals. The proteins are typically lysosomal or secreted, and proteolytic cleavage of the propeptide is required for enzyme activation, although bleomycin hydrolase is cystosolic in fungi and mammals. Papain-like cysteine proteinases are essentially synthesized as inactive proenzymes called zymogens with terminal propeptide regions. The activation process of these enzymes includes the removal of propeptide regions, which serve a variety of functions in vivo and in vitro. Amino acid residues within the pro region mediate their membrane association and play a role in the transport of the proenzyme to lysosomes. Among the most notable features of propeptides is their ability to inhibit the activity of their cognate enzymes, and that certain propeptides exhibit high selectivity for inhibition of the peptidases from which they originate. Isolation of papain. The full grown but unripe papaya fruit is subjected to shallow incisions on the four sides. The latex flows freely for a few seconds but soon coagulates. After collection, the coagulated lumps are shredded and dried by the sun or by the use of artificial heat. Incisions and collections are made at weekly intervals as long as the fruit excludes the latex. The fruit pepine is purified by dissolving in the water and precipitating with alcohol. From this picture, we can identify the unripe papaya from which the latex are collected. The incisions are made on four sides and the latex are collected from the unripe papaya. The chemical constituents. Papain contains several enzymes, peptidase capable of converting into dipeptides and polypeptides. A renin-like coagulation enzyme that acts on the casein of milk. An amylolytic enzyme which converts carbohydrates molecules into simple units. These are the chemical constituents present in the papain extraction. The functions of papain. The mechanism by which papain breaks peptide involves the use of catalytic triad with a cysteine. This cysteine then performs a nucleophilic attack on the carbonyl carbon of a peptide backbone. This forms a covalent acyl enzyme, intermediate and frees the amino terminus of the peptide. The enzyme is deacylated by a water molecule and releases the carboxy terminal portion of the peptide. Papain is a relatively heat resistant enzyme with an optimal temperature ranges from 60 to 70 degrees Celsius. From this picture, we can identify the process of flowchart of papain extraction. First, the collection of latex, then cleaning and sieving of latex, then mixing and drying, then packaging. Thus, they have developed the technical flowchart for papain extraction. The uses of papain. Papain breaks down tough meat fibers. Papain can be used to dissociate cells in the first step of cell culture preparations. A 10-minute treatment of small tissue piece less than 1 millimeter cube will allow papain to begin cleaving the extracellular matrix molecules holding the cells together. After 10 minutes, the tissue should be treated with a protease inhibitor solution to stop the protease action. Left untreated papain activity will lead to complete lysis of the cells. The tissue must be then triturated, passed quickly up and down through a pasture pipette to break up the pieces of tissue into a single cell suspension. Thus, papain is the main ingredient of paprika, a gel used for chemomechanical dental caries removal. It does not require drilling and does not interfere with the bond strength of restorative materials to the dentin. Papain has been known to interfere with the urine drug tests for cannabinoids. It is found in some drug detox products. Thus, the production of papain. The papain is usually produced as a crude dried material by collecting the latex from the fruit of papaya tree. The latex is collected after scoring the neck of the fruit, where it may either dry on the fruit or drip into a container. Thus, this latex is then further dried. It is known as a dried crude material. Thus, a purification step is necessary to remove contaminating substances. This purification consists of the solubilization and extraction of the active papain enzyme system through a government registered process. This purified papain may be supplied as a powder or as liquid. So from this video, we have learned about papain extraction, students. Thank you.